Alright guys, we're going to talk a little bit about DNA, a little bit about a little bit about RNA, and a little bit about DNA synthesis today. Okay, not quite as hard as it seems. We're not going to do too much on the history in here on this PowerPoint. We're going to do that kind of in class. So we will be leaving out a few key players in this PowerPoint, but we'll pick those up on an in-class assignment. Alright. Genes, DNA of some kind, either DNA or RNA, depending on bacteria, viruses, whichever, is in every living thing. Okay, when we talked about these characteristics of living things a while back, DNA, our genetic material, is part of that. Okay, so in 1953, Watson and Crick were given credit for the two-chained nucleotide joined by a nitrogen base. Okay, this is kind of the best example, is kind of like a twisted zipper. Okay, so if you had a zipper, well, the opener in the middle, if you kind of twisted it, all right, that would be a general structure of DNA. So here are two folks. These guys up here are Watson and Crick. Down here is Rosalind Franklin. She is one of my favorite characters, especially as far as, unfortunately, it's kind of getting misused and mistreated by science history. Rosalind Franklin's work was crucial to figuring out the structure of DNA, but in Watson and Crick's papers, when they presented their findings, she was not mentioned whatsoever. Unfortunately, she didn't really care necessarily. It wasn't really her thing. She wasn't worried about what DNA and how it looks. But still, just the fact that she was given zero credit till later down the road is still one of the great travesties, in my opinion, of science. All right, so DNA stands for deoxyribonucleic acid. All right, these are things that control every reaction. Well, we use our DNA and the RNA to build enzymes, protein structures, all these things that we will need to live our life. And it expresses, the reason Mr. Rogers has a red beard is somewhere in my DNA, all right, it codes for red beard hair, okay? All the instructions for manufacturing proteins and the enzymes and the structures and everything that you need is in your DNA. So the structure of DNA. The base unit is called a nucleotide. So if you remember back from when we talked about macromolecules, we talked about DNA and the subunit being a nucleotide. The nucleotide is made up of three parts. We have a sugar backbone, phosphate group that helps connect them all together, north to south, so to speak, and then the nitrogen bases, which helps us hold together left to right. All right, so we have four possible nitrogen bases. We have adenine, guanine, cytosine, and thymine. Now, due to Chargaff's work, he showed that there had to be equivalent amounts of A and T and G and C. So, if we have 50 A's, just for example, we have to have 50 T's. If we have 102 guanines, we got to have 102 cytosines, okay? Now, here's the reason is because, as you can see right here, we got 1, 2, 3 hydrogen bonds being formed between these molecules, and 1, 2 being formed between these molecules. So if we took a thymine and try to add it to a guanine, we would have like this whole section just, I don't know, floating around with nothing to bond to. So it just doesn't work. So once again, the backbone of this is the deoxyribose and the 1-phosphate, and then as always, A pairs with T and G pairs with C. So you can see here, okay, we have the sugar backbone. So here is a nucleotide. Let's just kind of mark off one. The P is for our phosphates. So the phosphates are how each one is bonded together. Like I said, north to south. The sugar is sitting in the middle. And then you have your nitrogenous base, all right, the A, the T, the G, or the C. So once again, A and T have 2 bonding sites, so that's why they bond together. G and T have 3, and that's why they bond together, all right? So you have 3 parts: phosphate, sugar, nitrogen base, okay? It's almost like I expect you to remember that. All right, so let's talk a little bit about DNA synthesis before we jump into RNA, okay? So in DNA synthesis, we have our parent molecule, the original strand. First, we're going to separate that strand, okay? Right here, as you can see, we're separating this strand. We have a molecule, DNA helicase. It's going to come down, all right, and unzip and unwind, or well, I should say unwind, then unzip the nucleotides from each other, all right, leaving them exposed, all right. So we have our base pair just sitting there doing its thing, but during S phase, all right, S, remember, stands for synthesis. We got DNA helicase comes along, unwinds, unzips, it leaves these edges exposed, all right. Then we come in and nucleotides are synthesized, all right, and then they are added complementary. So what that means is on this A, as we talked about, every A bonds with a T, and every T bonds with an A. Every C bonds with a G, every G bonds with a C, so forth and so on, okay? So what we end up having is 2 identical strands when all is said and done. So as you can see here, AT, CG, TA, AT, GC, boom, okay? Exact same thing over here. These 2 are identical. This is done with an efficiency rate of 1 mutation or 1 mess up every 1 in 1 trillion, okay? That's a lot, all right? So our DNA is pretty good at this. If you got 1 out of a 1 trillion, I'd give you an A+, because I'd have to, because that's an A+. All right, so now you have 2 identical strands left over, ready to go, ready to do their thing. Now we can move on, and we go, like if we're talking, prophase, we're rolling into prophase, then metaphase, anaphase, telophase, cytokinesis. Boom, we got 2 new cells. And here's kind of a visual representation. DNA helicase is going to be right here, kind of marching down the center, unwinding and unzipping this bad boy. All right, I'm going to leave RNA for another short video. I think that's enough for today. You'll have a good one, and see you in class.